Hi guys, welcome to this video. My name is Ms. Ngubane and I teach life sciences and natural sciences. Today I want us to do investigations related to the topic photosynthesis. These are light is necessary, chlorophyll is necessary, carbon dioxide is necessary, as well as oxygen is produced. All this is during photosynthesis. So these statements can be a hypothesis, an aim, or a conclusion. Now, at the end of each investigation, we do the Starch test, but this is except the last one, oxygen is produced during photosynthesis. So it's important for you to know the steps that are taken during the Starch test. So what you do is you place your plant in a dark cupboard. This is done to de-starch the plant so that the plant uses any stored starch. It is used during cellular respiration so that when you start your investigation, there's no starch present. And this proves that if there's any starch present at the end of the investigation, it was produced during the experiment. Now, the method to follow when you do the starch test, you need a Bunsen burner, a beaker with water. You place your leaf in boiling water. You do this to soften the leaf and also stop metabolism. You're going to then put your leaf in a test tube with ethanol and put it back in the Bunsen burner. We do this to remove chlorophyll by destroying the cell membrane, and this makes it possible for you to see the results when you do the starch test. Then you're going to remove your leaf, rinse it with warm water to soften it, and then add a few drops of iodine solution, and these will be your test results. Moving along to the investigations, we start with light is necessary for photosynthesis. So our aim here is to prove that light is needed for photosynthesis to take place. So you're going to de-starch your potted plant and then you cover it with a piece of foil. We're using foil here because it does not allow light to pass through. So this part that is covered is not going to receive light. Once you've done that, you'll place your potted plant in a sunny area. Remember that you're doing all of this while your leaf is still attached to the whole plant. Then once you're done, you're going to pick off the leaf and do the starch test. This is the results that you should be able to see. The part that was covered with foil should remain brown as the experiment, and the part that wasn't covered should turn blue-black to show that light is necessary for photosynthesis. So the part that is blue-black is our control. This is because the independent variable light was given to the leaf while the experiment we manipulated the independent variable. Our next investigation is to prove that chlorophyll is needed for photosynthesis to take place. Now here we are going to use a variegated leaf. A variegated leaf is a leaf that has two colors. One part is white and the other part is green. We do know that the green part contains chlorophyll and the white part does not contain any chlorophyll. In scientific concepts, they say that a variegated leaf has both the experiment and the control in one. So our experiment will be the white part and the control will be the green part, as it has starch while the experiment does not have any starch, since photosynthesis does not take place when chlorophyll is not present. In this case, we are not going to de-starch our plant. This is because we already know that the white part does not contain any chlorophyll, so there's no starch present there. So if you were to de-starch this type of plant, and then you do the starch test, all parts will remain brown. However, in this test results, the part that is green will turn blue-black, and then the part that is white will remain brown. Now let's do this past paper. It says a variegated leaf is characterized by irregular patterns of coloration ranging from shades of green to white. This variation results in areas of the leaf having reduced or no chlorophyll content, leading to lighter colored patches on the leaf surface. A variegated geranium plant was destarched for 24 hours. Then a strip of cardboard was placed over a leaf section on the plant and exposed to light for 24 hours. First question, you identify indicator A. We know that to do the starch test, you need iodine solution. Question B, chemical solution B. We know that you must put it in ethanol. Now you give letters of the parts in diagram C that will test positive for starch. We know part E is the white area and we must test negative. G and H, the dark cardboard prevents light from passing through. It also must turn negative. And only part F, the green area, will test positive. When you look at the mark allocation, it's only one area anyways. This means that we've identified E, G, and F as the parts that will test negative, and the question is 3 marks also anyways. Now for the next question, we arrange them according to the correct order. That will be C, B, D, and A. C being the start, B in alcohol, and D rinsing with warm water, and A where you put iodine solution. Now they say explain why solution B was used. That is alcohol, and the question wants you to explain, so you need to describe first that alcohol removes or breaks down the cell membrane that allows it to remove chlorophyll. Next question is asking why a variegated leaf was used. We know that a variegated leaf has two colors, the green part and the white part. So in this case, we're using it to compare the parts that are green and the parts that are white. You know that the parts that are green, they have chlorophyll, while the parts that are white do not have chlorophyll. So it's used as a comparison. Remember that explain questions require you to describe and give the why. So it's a 2-mark question. The first mark goes to compare and the other part will be where the other mark goes. Last question, you describe the effect of increasing light on photosynthesis. We know that as light intensity increases, the rate of photosynthesis will also increase, but up to optimum level, since any other factor affecting photosynthesis becomes a limiting factor. This sort of is a conclusion for the investigation that will be covered in part 2.